Hello. Today we're going to learn about DNA and its history. DNA is the body's blueprint. It's what makes us us. It gives us brown eyes or blue eyes, brown hair or blonde hair, and it also determines quite serious things at birth, such as if we'll have a genetic disease or we won't. It's very, very important to our bodies and to everything living on this earth. However, we did not know much about DNA at all before the 20th century. The history of DNA really started in 1928 with Frederick Griffith, who was a experimenting scientist who wanted to know about the genetic materials within cells, but didn't know what it was, so he was determined to figure out what exactly was that genetic material. What he first started doing was he took strains of Streptococcus pneumoniae, which is what we know as pneumonia, and he took two different strands. He took the smooth strand S and the rough strain R. Only the strain S was deadly, and he injected them both into mice. So what he first did was he had a mouse, two mice. He injected the smooth strain S into one mouse, and that's the deadly smooth strain S, and the rough strain R into another mouse. And the mouse that died was the one who was injected with the strain S. However, when they injected both strains after they had been heated and killed, they figured, well, they'll probably be harmless because we've killed the deadly one, and the rough strain R doesn't affect mice at all. They injected into one of the mice, and it ended up killing the mouse because it suddenly caused living strain S to appear. In conclusion, Frederick Griffith discovered that something from the dead S converted the strain R into living strain S. What this transforming principle was DNA, and that's what Frederick Griffith was looking for. But he didn't quite just figure out what exactly DNA was. Next, we have to go to Avery, McCarty, and McLeod's experiment, which happened in 1944. And that was when we really found out what exactly the DNA was. The experiment began with Oswald Avery, a molecular biologist, and his colleagues Macklin McCarty and Colin McLeod. They followed practically the same experiments of Frederick Griffith, and were seeking out to discover what exactly the transforming principle that he found was. Now, during this time, around 1944, many people, scientists as well, believed that DNA was a protein or a carbohydrate, something simple. This team of scientists, however, proved them wrong by discovering what exactly this transforming factor was. What Avery, McCarty, and McLeod set out to do was to purify 20 gallons of the same Streptococcus pneumoniae, or pneumonia bacteria, for this transforming factor that Griffith had found. What they did was they heated the bacteria and then filtered it out multiple times using different alcohols and chloroform and other things until they could physically see the strands of the bacteria. And when they went in to focus in on this transforming factor that they had pulled out from the bacteria, they found that it was indeed not a protein nor carbohydrate, as most people had suspected it was, but the nucleic acid that we now recognize as DNA. Because many people still believed that DNA was too simple to be this nucleic acid that Avery, McCarty, and McLeod found, their experiment and their results were met with very much skepticism, and so many people did not believe what they had found was really the answer to what DNA was, unfortunately. The next experiment that took place was Hershey and Chase's experiment in 1952. They worked with things called phages, which were small bacterial infecting viruses that had an outer protein coat that surrounded DNA. Because Hershey and Chase knew that the phages were transferring hereditary information to the infected bacteria, they decided to make an experiment based off of that so that they could determine from the phages whether or not it was the protein coding that was carrying the information or if it was the DNA that was carrying the vital information. The experiment they conducted in order to determine this was that they infected bacteria with the viruses that had either radioactively labeled proteins or DNA. So some of them had protein that was labeled and some had DNA was labeled. So the infected bacteria became radioactive only from the DNA, not the protein. So it concluded that DNA was the molecule of heredity. The next experiment was conducted by Franklin and Wilkins in 1951 through 1953. All of the experiment really began with Maurice Wilkins, who in the late 1940s began to use optical spectroscopy, which was a means of studying the properties of physical objects based on measuring how much the object emits or interacts with light. And he used it to study DNA. So he really focused on honing in on using the spectroscopy. And then in 1950, he was actually able to make a crystalline X-ray. So it was like a crystal X-ray, and to see the diffraction patterns clearly of the DNA, but he wasn't really able to really see the structure or to recreate the structure into a 3D model of DNA. But then comes in Rosalind Franklin, who was a chemist and expert at X-ray diffraction, which is a technique that uses focused beams of X-rays to produce photographs of the crystallized forms of molecules. So, because she was able to take the crystallized form of the molecules that Wilkins created, she was able to, at the end, really truly discover the 3D model of DNA. So what she did was she X-rayed the crystal, and when the light passes through the molecules, they diffract, so they bounce off of each other, and then strike onto the photographic film she has set up. And because they bounce on the film, different patterns were produced that could then be used to deconstruct and then reconstruct the 3D dimensional structure of the DNA molecule, therefore revealing its exact structure. The information that was found by the experiments done by Franklin and Wilkins was actually taken and interpreted by a different set of two scientists who were named Watson and Crick. The interpretation by Watson and Crick began in 1953 when they viewed one of Franklin's X-ray diffraction images without her knowledge and figured out exactly what DNA was made of and how it was built up. They figured that it was two mirror image strands composed of stacked DNA nucleotides, a sugar of phosphate plus a nitrogen base, and were linked together into a double twisted helix. The double helix, which is formed from the stacked nucleotides, resembled a ladder. The sides are made from the deoxyribose sugar and the phosphate, and the rungs, which go across, was the adenine, the thymine, the cytosine, and the guanine, that were all joined with weak hydrogen bonds. We learned that A always binds to T, and C always binds to G. And the order represents our genetic sequence. So how they fit up and how they match, well, they always match the same, but their order that they're in determines what we'll look like. Because Watson and Crick actually interpreted this information. They were awarded the 1962 Nobel Prize in Chemistry, and because Franklin died in 1958 from cancer, she didn't get the Nobel Prize, which actually made me a little upset because it's not given posthumously, which means it isn't given after death, but that just made me a little upset that she didn't get it. The last major experiment conducted for DNA was the Michelson and Stahl experiment in 1958. They used the DNA model that was interpreted by Watson and Crick and worked from it to experiment on how DNA replicated. They figured that since the double helixes of DNA were complementary, and they worked from it as a template to make new complementary strands. They determined 3 models that could possibly work. One would be a resulting helix that contained an old and a new strand, called semi-conservative replication. The next model would be the old DNA is preserved and an entirely new strand is created, called conservative replication. The third model would be 2 molecules with old and new DNA interspersed along each strand, called dispersive replication. Now, they wanted to prove that the Watson and Crick model was correct, and in order to do this, they had to distinguish between the old and new DNA. So, they altered the density of 2 strands of DNA. They added N15 nitrogen to one strand and N14 nitrogen to another strand, and then placed them both into a centrifuge and spun it around. They determined by altering the density and using the centrifuge that it was semi-conservative replication, not the other 2 models. So, DNA would replicate by making a resulting helix with one strand of the old DNA and then an entirely new strand. So what it would do is it would break apart. You've got one helix, and then you would break apart the 2 strands. So if you had an A on this side, you would have to have a T on the other. You would break them apart. Then one would have the old T, and then it would make a new A. This one would have the old A and it would make a new T. And that, again, is called semi-conservative replication, and that is how DNA replicates. So now we're here today, after all of these really fantastic, extremely revolutionary, and very insightful experiments, and we know today that from all these experiments exactly what DNA is, that it's not a protein or a carbohydrate, that it is a nucleic acid, that it's a double helix structure with rungs that are made up of A, T, and C and G, and that it's our genetic material. It's what makes us us, and it's extremely necessary to our world today.